this is part 2 of our immunology review. We recommend that you watch part 1 first, where we talk about cells of the innate immune system. In part 2, we're going to be looking at cells of the adaptive immune system and how that works. So just to recap, cells of the innate immune system include natural killer cells, macrophages, dendritic cells, and granulocytes such as neutrophils and eosinophils. Cells of the adaptive immune system involve T cells and B cells. And the main difference you want to remember is that innate cells recognize a limited number of antigens, while cells of the adaptive recognize millions of antigens. Before we go on to talk about the adaptive system in more detail, it's important to know the difference between primary and secondary lymphoid organs. So primary lymphoid organs are where immune cells are produced. That involves B cells and T cells. So there's the bone marrow and there's the thymus. Secondary lymphoid organs are those where the immune response actually occurs, and that involves the lymph nodes and the spleen. So how does the adaptive system recognize pathogens? Just a reminder that the innate system recognizes pathogens with pattern recognition receptors and pathogen-associated molecular patterns, but we're now talking about the adaptive. So it starts off with a dendritic cell picking up parts of the dead pathogen. The cell becomes activated and mature and processes what it's taken up and chops that protein up and presents it on the MHC molecule, which we talked about in part 1. It then goes to the draining lymph nodes and presents the antigen to T cells to check if they recognize it. MHC class 1 molecules, which are present on every nucleated cell in the body, present it to CD8 cells or cytotoxic cells, whilst class 2 present it to CD4 cells, also known as helper T cells. So what's the action of cytotoxic killer cells? They basically kill the cells via apoptosis. CD4 cells, they differentiate into various types of effector T cells depending on the cytokines present. And in case you didn't know, cytokines are basically just small proteins of the immune system involved in cell signaling. So there's 2 pathways it could go down. So in the presence of IL-12 and IL-13, both examples of cytokines, it differentiates into Th1 cell. In the presence of IL-4, it differentiates into a Th2 cell, which also produces IL-4. For example, mast cells make a lot of IL-4 and they respond to worms and multicellular parasites. Th1 cells are involved in cell-mediated immunity, whilst Th2 cells are involved in humoral immunity. Cell-mediated is where you're talking about intracellular pathogens, whilst humoral is extracellular, and that involves B cells. And so let's talk about B cells. The B cell receptor is actually an antibody on the surface membrane. And it's important to know that the antibodies are only secreted when an infection is detected, causing B cell activation. But what leads to this B cell activation? So you have your T cell, and it recognizes fragments of antigen via the MHC molecule. It recognizes it and it becomes activated. And then a B cell antibody recognizes that same pathogen via linked recognition between the T cell and the B cell, and that in turn activates the B cell, so it starts producing more of the antibody and then secreting it. So antibody structure, we'll just talk about that quickly. So the antibody structure involves 2 heavy chains and 2 light chains. It's got the FAB region, so the antigen binding region, and FC, or the constant region, which determines the function. There's different types of antibodies. So monomers are IgD, IgE, and IgG, which is the most common. Then we've got dimers, which is the IgA antibody, and finally pentamer, which is the IgM antibody. Going back to functions of antibodies, so different antibodies act in different ways. You have neutralization, agglutination, opsonization, antibody-dependent cell cytotoxicity, as well as activation of complement. It's important to know that CD4 cells can also become other types of cells, so not just Th1 and Th2. They can also differentiate into Th17, depending on the cytokines present, and that helps in defense against extracellular bacteria. And in the presence of TGF-beta, it can differentiate into a T regulator cell, which secretes TGF-beta and helps in immunosuppression. A fraction of the activated T and B cells become memory cells, providing long-term immunity to that infection, and they are stored for rapid deployment if infection reoccurs. Thank you very much for watching.